evolution of mutagenesis.
From the board there, people from 12 countries are here, including India. There are only two things in the world which brings people together. The world is very chaotic, but we bring the people, science brings the people together. And you will be bringing them together when you will be organizing or you will be collaborating with the people outside India or outside your lab. So, Dr. Prasad asked me to give a talk and I wasn't sure what to say or what topic I should choose. And I saw my name was on the top, so I suggested that I use this title, Mendel to Mutation, Origin, Evolution and Significance of Mutagenesis. Some of the information I am giving you, you might already know. So it will just refresh your memories. But it should bring the things in the context. Okay, so as we know, Mendel is considered to be father of genetics. But he didn't talk about the genes or chromosomes, anything. He talked about factors. And we also know that the two laws he proposed, law of segregation and law of independent assortment, his hypothesis was what happens if two contrasting pair of characters, they are in hybrid, what happens in the next generation. And he found out that they segregate and they segregate independently. He presented his work, two papers, in 1865 in Broome Society of Natural Sciences. Why this is important? If he would not have presented his work would have been lost from the scientific world. So it is important whatever you do, you present it or you write it. And that's how it gets recognized. His law was not, because people get lost with all his mathematical explanations, and he said, my scientific studies have afforded me great gratification. Those days scientists enjoyed doing science. And I am convinced that it will not be long before the whole world acknowledges the results of my work. So his laws were not recognized when he was alive. What happened then? In early 1900s, these three scientists, Hugo Frisch from Holland, Karl Koretz from Germany, and Tesmark from Austria, they independently discovered Mendel's laws. They used plant. He used evening primrose, Koretz used maize, and he used pea plant like Mendel. And independently they came to the same conclusion. And remember, Mendel didn't talk about the genes or chromosomes, he talked only factors. The term gene was coined by Johnson, it was Danish pharmacist in 1909. And genetics, English biologist, was coined in 1906. But the term mutation was coined by Hugo Frisch. He was working on evening primrose, very tiny little plants you will find in Europe. And mutation, he saw in the evening primrose was actually not the mutation, it was Renner's configuration, rearrangement of chromosomes. But he coined the term mutation. So they all worked on plant. One more scientist who died quite early, Theodor Boveri, in Würzburg in Germany. Würzburg has Germany has produced many good scientists. X-ray was discovered there by Röntgen. Rudolf Virchow discovered the inflammation, and Boveri, along with Walter Sutton, he proposed chromosomal theory of inheritance. It is the chromosomes which go independent assortment and segregation, what Mendel had proposed. But the important point Boveri also proposed was the origin of malignant tumors, and he used sea urchin embryos as his model. And in Wurzburg, I go often there. His statue is there, microscope. These days we have so many sophisticated microscopes. This is his microtome. And this is the diagram he used to draw, how chromosomes go to different parts to form malignant tumors. So role of chromosomes in tumors was established by Boveri. Also chromosomal theory of inheritance. Unfortunately, he died at a very early age, 52 years since he died, and people suggested that he might have won the Nobel Prize if he would have been alive. If you see this article published in Nature Reviews in 2001, Boveri had predicted so many other things: cell-cycle checkpoints, tumor suppression in oncogenes, tumor progression from benign to malignant, genetic mosaicism, clonal origin of tumor. All these things he had already proposed that these are the things that will happen or this is happening. Go back in early 1900. So he was a very remarkable scientist of his time. Now, people until early 1900 were working on plants. Awadhesh, Correns, Tesmar, and other people, they were working on plants. But the real breakthrough came from Thomas Hunt Morgan, T.S. Morgan, in Columbia University. He was basically an embryologist or developmental biologist. He was working on Drosophila. And in his Drosophila stock, he found one male fly with white eye. Normally they have red eye. One male fly from thousands of flies, that was white eye. And he became curious. And that was a mutant. And through breeding that white-eyed fly, he discovered that genes are linearly arranged on the chromosomes. Chromosomes, they are important, and genes are linearly arranged on the chromosomes. And he got Nobel Prize in 1933. And he switched from embryology to genetics because chromosomal theory of inheritance was being established. He was remarkable. He wrote 22 books and over 370 papers. Established Drosophila as the model species to work with. Very, very important. So he is considered to be father of fruit fly genetics. He produced many students, good students, and one of them was Muller, H.J. Muller, Hermann Joseph Muller. And X-ray was invented in 1895 by Röntgen in Wurzburg, but no one knew the impact of environmental agents like X-rays on genes. And remember, you have a photo on your corridor. DNA is the genetic material was established in 1944. So Muller, through his experiments with fruit flies, wrote a paper, Artificial Transmutation of the Gene 1927, for which he got Nobel Prize in 1946. X-ray can induce mutation. Very interesting life of Muller. If you find it, it will inspire you. He worked in many places, in Germany, in Russia, in UK, in America. And there was a review article his student wrote, was there any life before 1953? So Muller was not only saying that X-ray can induce mutation, but he also gave many ideas. He was a philosopher. Because people were at that time searching what is the genetic material. And Muller gave the idea that whatever is the genetic material, protein or DNA, main targets, they should be able to replicate itself or make their own copy, all those things. So that's why this article is very important. Was there life before? So his contribution. After Muller did it in 1927. He was based in Edinburgh, in Scotland. One of his students, S.P. Roy Chaudhary, was from India, Banaras University was here. I know him from my childhood. He died in 1994. My PhD supervisor and my father did PhD with him. So when Muller gave his Nobel lecture in 1946, based on his experiments, he said no escape from the conclusion that there is no threshold. There is no threshold value for X-rays or any carcinogenic chemicals. So when a single cell gets mutated, if it is not repaired, because DNA is the only material which can repair its damage, it could lead to malignancy or tumor. So there is no threshold. And that principle also applied for chemicals which still exist, although people were trying to say low dose is good, hormones and helps, but there are lots of controversy. So that Nobel lecture was based on the work which Roy Chaudhary did during Second World War in Edinburgh. And Roy Chaudhary also contributed a lot. He was a good friend of all the big biologists of his time. J.B.S. Haldane, M.J.D. White, Alfred Grob, Dr. Jansky, all were his friends. So that was X-ray. Muller's student, Charlotte Orbach, she was also working in Edinburgh. And during World War II, she had come during World War I from Germany as a refugee. So she used mustard gas or nitrogen mustard. So it's not only X-ray, but chemical can also induce mutation. But her work was not published until the Second World War was over. So these were the founding fathers of mutagenesis. Although the work in plant continued for some time, Carl Sachs and other people, they worked on plant species, but then eventually moved to Drosophila and other mammalian species. I hope I am making myself clear to you. Yeah. So what the mutations they do? Mutagenesis plays an important role in our lives. A low level of mutagenesis is advantageous and ensures the survival of species by promoting evolution. We know that mutation is important for evolution, but you have to remember, individuals don't evolve. It's the population which evolves. Individuals only adapt. and evolution takes place thousands of years to evolve. So it's the change in the gene frequency. Programmed and mutagenesis of immunoglobulin genes promotes diversity and provides dynamic defense against invading pathogens. How we adapt with coronavirus and all those things, it's our genes. Many human diseases, including most cancers, I will come back to that in a second, that is important, arise because of mutations and accumulation of oxidative agents, changes in the genes because of oxidative stress, generation of free radicals or reactive oxygen species. They also are implicated in premature aging and neurodegenerative diseases, Parkinson and Alzheimer's. Mutagenesis also drives the evolution of virulent bacteria and viral pathogens, allowing them to evolve resistance to therapeutic and thwart the immune response. So these things probably you know. Now, cancer. What we know about mutations and cancer? As I said earlier, mutation plays an important role in our lives. What we know about cancer, 3 facts about the cancer. 95% of the cancer is triggered by the environment. Only 5% of the cancer is inherited. Some of the breast cancer or prostate cancer or some childhood leukemia, 95% is through environment. The second is current rate, at least 50% of the people in Western world will die with cancer. That's the, and it could go even more, despite the fact that science is making progress in diagnosing and treating them. But it's going up. We have not been able to. And what was the third? So 95% this and what? I will remember and come back. So you can see mutation plays an important role in all aspects of life and it affects aging, cardiovascular. So mutation plays a very important role in our life. Some of the mutations, they are expressed early in the life, like fetal wastage, spontaneous abortions or induced abortions. That is also a type of mutation. And many, they appear very late in the life. Coronary heart disease, schizophrenia or sexual orientation, all these things, they all have some component of mutations there. So that's what I was saying here, teratogenesis and decreased fertility. So we talk about SNM, oxidative stress, generation of free radicals. That could be either through external agents, these are the agents, irradiation, ionizing radiation, metals, metabolism, and then endogenous through metabolic processes when we generate reactive oxygen species. They all damage DNA and they can lead to a range of diseases, although reactive oxygen species are implicated in many diseases. How to prevent oxidative damage in whole organism level is not very clear. How we can do that? So mutations we can classify as a microlesions, MICRO, or macrolesions, base pair substitution, MC mutations, or structural changes in the chromosomes, or numerical changes in chromosomes. But numerical changes in chromosomes, that's why I have highlighted in different color. This is because of malfunctioning of microtubules. It's not damage to the chromosomes. So Mutations can occur at base individual gene chromosomes or total genome. We change the chromosome number. And 50% of the mutations, this is based on Professor Shankar Narayanan, the great son of Tamil Nadu. 50% single base changes, 50% deletions, duplications, insertion or translocations. This is based on molecular basis of inherited spontaneous mutations in over 100 different human genes. 2 points here to remember. We already know what is mutation. Don't have a chalk here, I would have explained it, but don't worry. Mutation is inheritable changes in the genetic material. Thank you. So here you have a cell, and here it is DNA, right? And you induce a damage here through different agents, and it is only mutation only when this damage is transmitted to daughter cells or to the next generation through germ cell. Inheritable changes in the genetic material. Mere damage is not mutation. And the second thing is, all the mutations or all the damage to DNA is subject to repair, DNA repair. DNA is the only material in the cell which is capable of repairing its damage, right? So most of the mutations, they occur because of faulty DNA repair. Am I making sense to you? So that's important to remember. So this is from Professor Natarajan and Gunther Away. What are the molecular agents in the DNA? These are alkylation. Chemicals transfer their functional group, alkyl group, ethyl, ethyl, propyl. A-pyrimidinic site, loss of pyrimidine. Phosphotriesters are monofunctional alkyl agent. A-purinic site means loss of purine. Intercalation like acridine orange, which will create femcicylation. Single strand breaks, bulky DNA attacks through polycyclic aromatic hydrocarbons. Base damage, double strand breaks through different chemicals, especially in the radiations, violet radiations, inter-strand crosslinks, intra-strand crosslinks. So these are the lesions at the DNA level. Most of them they are repaired, but most difficult is the double strand break repair. If they are not repaired, they will lead to mutation. Mostly if double strand break is implicated in chromosomal arrest, which are implicated in initiation and promotion of cancer. This is for radiation. So at molecular level, further to that, there are 17, these are the bases, purines, guanine and adenine, pyrimidines, cytosine and thymine. There are 17 nucleophilic sites in the DNA, which are DNA bases which are vulnerable to damage by different agents. And each atoms have different charges, negativity. The nucleophilic substance have extra electrons, and electrophilics, they are short of electrons, so they attack to get excess electrons from them to make them unstable. I'm sure you have learned all these things, yeah? And these are some of the characteristic transition and transversion by different agents like benzoylpyrene, they mostly lead to G2P transversions. DNA repair is highly conserved processes. There are recombination repair, mutation, non-homologous indisjoining nucleotides, repair, base repair, direct transversal, mismatch repair, and you can see they are overlapping with each other, and they have common mechanisms: cut and paste, recognize the damage, cleave it, DNA polymerase will come and ligase them with DNA ligase. And there are at least 200 genes, highly conserved genes, which are involved in DNA repair process, right? And they do other activities as well, for example, DNA replication process. And these are important genes: BRCA1, ATM, CHEK2, ATR, P53, which is the master gene. So they are important. That, and this is hallmark of cancer. So it's not only mutations which is playing important role in malignancy. You are enabling replicative immortality, inducing angiogenesis, formation of new blood vessels to supply oxygen and nutrients to tumors, resisting cell death, sustaining proliferating, signaling, evading growth suppressors, activating invasion and metastasis. So this is a very important paper by Hahn and Winberg. Winberg's paper are very important in developing these concepts. But I thought I should mention this to you. This is available open access as well. So you can find fighting immune destruction, tumor promoting inflammation, genomic instability and mutations. So these are very complicated mechanistically processes. And scientists are still trying to understand what leads to how a cell emancipates or becomes selfish to form a tumor. But we are not able to understand that. Every month or every 2 months you will find a new gene or a new protein being discovered which is involved in malignancy, but still we don't know the mystery of life here. So then, from mutation and cancer, public and regulatory concern, government thought, oh, we have to do something. If environmental agents are inducing cancer. So early mutagenesis studies used Drosophila plants continued for some time. Even the United Nations developed environmental development programs, they promoted plants, but it's in decline. Fungi. It was not until the 70s that the mechanisms of action of mutagens and carcinogens were linked, mainly due to the development of exogenous metabolic systems, we call it S9 system, mimic the activity of mammalian liver. And this paper by Bruce Ernst in 1973, carcinogens are mutagens, that changed the whole concept of how we can test the chemicals for their mutagenic potential and carcinogenic potential. So mutagens are also carcinogens. They are also clastogens. They are also teratogens. And they are also carcinogens. So that led to a series of different test systems. By the end of 1970s, at least 40 different test systems were in use. Mutagens and therefore potential carcinogens were being identified in many materials, including food and drink. I will come to that in a moment. So this started, and UK took a leading role in this one. I will come in a moment. So it was impossible for government and those responsible for human safety to develop any guidelines when so many test systems were in use. So the first collaborating program was established by John Ashby and Fred Viseris from USA and UK. John Ashby did a lot of work on this. And carcinogen and non-carcinogen chemicals with similar structure. Only about 4 or 5 test systems emerged from this trial, and any reliable performance at distinguishing between carcinogens and non-carcinogens analogs were confirmed. These form the basis of the battery of tests recommended in various regulatory guidelines and are still the basis of testing strategy. Dr. Kumar Ravel can tell you more about that. And David Kirkland, John Ashby and Brian Bridges from MRC from UK. UK scientists took a very leading role in this tiered approach. So first, EMS test in vitro screen for genotoxic potential. If it is negative there, in vitro cytogenetic assays, cell-based culture. If it is also negative there, if it is also negative there, regard this as non-genotoxic. But if it is positive in EMS test, positive in cytogenetic, in vitro cytogenetic assay, then you treat this as in vitro genotoxic. Then you move to in vivo system, bone marrow micronucleus assay, or liver DNA repair assay, or UPS assay. If they are negative in both of them, you regard them as a possible carcinogen. If not, then regard them as a non-genotoxic. But this is very complicated process and very expensive too. So this liver DNA repair assay eventually got replaced with in vivo comet assay. And in this process, International Workshop on Genotoxicity and Test Procedures, IWGTP, every 2-3 years, David Kirkland from UK used to work with Cohans, now he has independent status. So he organizes this workshop inviting leading workers to contribute to that. So there have been further improvements in the recommended test systems, highly sensitive scientists. Some of these were now being non-genotoxic carcinogens, how to use them. Non-genotoxic carcinogens, they don't induce mutation as such, but proliferation and other aspects are there. So they keep on modifying these protocols there. So I'm conscious of the time, but most of the informations are there. So the genotoxic test we use today have changed little over the last 30 to 40 years. Some have been made more extensive, and in some cases top limits for testing have been reduced. But many features of the test, including cell types, measures of toxicity, exogenous metabolisms, have not changed much. So this area is still developing, and where industry, the regulators and the scientists, they come on one platform and discuss how we can improve it further, make it less expensive and more robust. And they do a lot of trials as well, collaborative trials. This I thought I should also mention here, because we often consider that chemicals and radiations are harmful to our health and reduce nutrition. But food is also part of our environment. Diet or food habit is related with cancer incidence, and it is suggested nearly 40% of the cancer is because of our food habit. There are some good food, some bad food, aflatoxin here. You will hear from Mike Douglas about aflatoxin in the conference. Some food which are cooked at high temperature, producing nitrosamines, or red meat leading to colon cancer. A lot of work has been done. And Hippocrates, father of modern medicine, if you go to Fossil Island in Greece, you will find a museum there. Let food be the medicine and medicine be the food. So these are One aspect is mineral levels in meat and milk plummeted over 60 years. Minerals, micronutrients we need in our food, and they are declining. And these are some of the good food there, but we have also concerned with highly processed food and drinks, energy drinks or fizzy drinks, all those things. They are very much concerned. So micronutrients, they are plummeting in our food, and this is Bruce EMSI, passed away last year. Nina would know, 95 years of age, he passed last year, but from EMS test, he moved to this type of activities. One of the titles of his paper was DNA damage from micronutrient deficiency is likely to be major cause of cancer. That was the title of his paper, which was published in Nutrition Research. So what the micronutrients do? Micronutrients are vitamins, essential minerals and other compounds. There are 40 required in our diet, and they play a role in DNA maintenance, DNA synthesis, repair, methylation, chromosome breakage, segregation. And if they are deficient, if our cells are deficient, the deficiency can mimic radiation or chemicals damaging DNA by causing single strand breaks or DNA double strand breaks. If there are not enough micronutrients there, DNA synthesis, repair, methylation, all will be impacted. And that will lead to single strand breaks or double strand breaks. So in that cell, mechanisms of human carcinogenesis, environmental carcinogens, our lifestyle. After Second World War, people started going to sunbathing, holidays, smoking, drinking. That is all our lifestyle, good food and bad food, but also our genetic makeup. How our genetic makeup respond to the environmental agent. And then cancer. Cancer, remember, this is not happening in 1-2 days or 1-2 years. It's a latent process, 20 years, 30 years time. And cancer originates accumulation of mutations in single cell. In single cells, you should have 6 to 12 mutations in critical genes. Then only the tumors develop. So, if anyone has got a cancer induced by environment, you must have been exposed to that chemicals or agent 20-30 years ago. So now, as I said in my inaugural remarks there for this workshop, science is becoming more and more interdisciplinary, multidisciplinary. So you can see physics, chemistry, computer science, biology, engineering, they all playing important roles. Genomics, transcriptomics, proteomics, metabolomics, liposomics. And then here you get ethical measurements. Ethical measurement is indicator of adverse effect on the organ level or individual level. Now we have computational models. AI techniques are being used there. And the aim is to have safe drugs, environmental protection, green chemistry, and safe food. So, finally, this is the concluding session. The science of genetics and mutations has progressed a long way since the discovery of Mendel and other scientists. Radiation and chemically induced mutation and cancer is still a serious concern and need to be avoided. The tests being developed and used to predict genotoxicity, but their specificity is often being questioned. The genotoxicity test being used today have changed little over the last 30-40 years. Science is becoming more and more interdisciplinary, driven by new technologies helping to elucidate the mechanisms of mutagenicity and carcinogenicity. omics, epigenetics. I did talk about epigenetics. That's an important aspect there as well. And emerging technologies, artificial intelligence or machine learning will play an important role in elucidating the mutagenic and genotoxic impacts on our genome. So I think with that I will stop, but I will be happy to answer if you have any question. Thank you very much.
Your elucidative lecture. The audience may ask questions.
Yeah, I am happy to answer. If not now, I am here for next 2-3 days. I will be happy to answer questions. So think about that, but I am happy to take questions. Any question? No? Yes, Ruth.
So, this is essential for health and for evolution. Why do you think that genomic instability wasn't in the first set of hallmarks of cancer by Hannah and Weinberg? Sorry, it's a loaded question.
I think they had mentioned something, genomic instability.
It wasn't in the first six. They put it in the
Second one.
They put it in the second one. They didn't include it in the first one, and it feels like a fundamental.
Yeah, I was surprised too, but I thought maybe it was in the second one, not in the first one.
Yeah.
Whereas it should be, given the fact that people like Bovary proposed long time ago that stability is important for development of cancer. So yeah, and maybe he rectified in his second paper. Same question could have been asked to him and then he rectified it. Thank you.
Can I ask?
Yes, Dr. Rath.
Do you still believe in LNT theory, linear no threshold?
This is a big debate. LNT theory. If you believe, Calabrese, he will say hormeosis. And in Swansea meeting, you remember Calabrese was there. And he was against Muller and Raichaudhuri, he was saying there. And Errol Jagger and George Hoffman, after his lecture, he had 1 hour training. And he was told that Raichaudhuri and Muller is not here to defend themselves, and your hormeosis theory has no experimental explanation. So LNT theory, even theoretically, USCPA, that's what it believes, that if one cell has got one cancer and if it is not repaired, then it is possibility that will get developed into cancer. So that's what the EPA is still believing. So my opinion will be, we have to adopt precautionary principle rather than weight of evidence. If you have to protect, if you are in the government, you need to protect the health of the population, and you have to do the precautionary principle, not the weight of the evidence principle.
No, I was asking from a different perspective.
Yeah.
Low doses, why it is so difficult to extrapolate?
Yeah, it's
I'm not trying to the
it's very difficult. It's the dose response curve at the low dose, it is still whether it is like this, that or that. When I was doing my PhD, I was in Banaras and I was involved in IAEA project of low dose risk estimation, and at low dose it's very close to background level, and background level is so variable. It's very difficult to, especially with the chromosomal aberration level, it's very difficult to establish that. That's my, I don't know if I answered your question.
We will have it.
Okay, thank you. More, when I was a student like you, I used to ask a lot of questions. Yeah. I tell you that the story in Banaras where I did my PhD in cytogenetics lab, we had seminars every week, and we had no PowerPoint, no transparencies on whiteboard, and we had 3-4 hours grilling by teachers. 30, 40 people sitting in one room. And we had lots of that training would help us a lot in future. So participate and ask questions. Questions can never be wrong, but don't hesitate. Any question? No? Okay, thank you very much. Yeah. Thank you very much. Take your photo.